The biosynthetic pathways leading to amino acids and nucleotides share a requirement for nitrogen. However, 4/5 of molecular nitrogen exists as atmospheric nitrogen inaccessible to most living organisms. In this video, I'll cover the nitrogen cycle and the regulation of nitrogen metabolism in our body. The nitrogen cycle involves pathways by which nitrogen from the environment is introduced into biological systems. It includes three main steps: nitrogen fixation, nitrification, and denitrification. Only certain bacteria and archaea can fix atmospheric nitrogen, including the nitrogen-fixing bacteria that lives as symbionts in leguminate root nodule. Nitrogen fixation is carried out by a highly conserved complex of proteins called the nitrogenase complex, found in the anaerobic legumoglobin. Its central components are dinitrogenase reductase and dinitrogenase. The net reaction involves the reduction of atmospheric nitrogen to ammonium, a change in the oxidative state of nitrogen from 0 to -3. First, 8 electrons are transferred from pyruvate to dinitrogenase via pyridoxine or flavodoxine and dinitrogenase reductase one at a time, producing 4 acetyl-CoA and 4 carbon dioxide. The hydrolysis of 16 ATP molecules are required to bring about conformational changes to overcome the high activation energy of nitrogen fixation. Fixation of each nitrogen molecule requires at least 6 electrons, and the 2 additional electrons are used to reduce 2 protons to hydrogen molecule in this process. Although ammonia can be used by most living organisms, soil bacteria derive their energy by oxidizing ammonia to nitrite with the oxidative state of +3. And nitrate with the oxidative state of +5. Therefore, nearly all ammonia reaching the soil is oxidized by nitrate. Finally, the nitrifying bacteria reduce nitrate to atmospheric nitrogen under aerobic conditions, completing the nitrogen cycle. The nitrogen cycle is short-circuited by the ammonox bacteria, which converts ammonia and nitrite to atmospheric nitrogen. First, hydrazine hydrolase converts ammonium and hydroxylamine into hydrazine and water. Hydrazine is a highly toxic molecule that is sequestered in a specialized organelle known as N-amoxisome. The membrane of this organelle is composed of lipids known as ladder rings, which contains few cytobutane rings that form a very dense barrier to slow the release of hydrazine. The hydrazine oxidizing enzyme then oxidizes hydrazine to generate protons and atmospheric nitrogen. On the surface of N-amoxisome, nitrate-reducing enzyme converts protons into hydroxylamine and water, completing the cycle. The ammonox bacteria offer a major advancement in waste treatment, reducing the cost of ammonia removal as much as 90% and reducing the release of polluting byproducts. Humans and most animals utilize nitrogen in the form of ammonium. Vascular plants convert nitrate to ammonium in a 2-step process known as nitrate assimilation. First, nitrate reductase catalyzes the 2-electron reduction of nitrate to nitrite, in which NADH serves as the electron donor, passing electrons successively through thiol group FAD, thiochrome B557, a molybdenum-containing cofactor, then to nitrate. Next, nitrite reductase catalyzes the 6-electron reduction of nitrite to ammonium, in which pyridoxine serves as the electron donor, passing electrons successively to iron-sulfur center, heme, and then to nitrite. The reduced nitrogen in ammonium is incorporated into biomolecules through 2 amino acids, glutamate and glutamine. Glutamine synthetase incorporates the nitrogen in ammonium into glutamate, producing glutamine, which is coupled to the hydrolysis of ATP to ADP and inorganic phosphate. The amide nitrogen of glutamine then serves as a source of amino group in a wide range of biosynthetic processes. Since glutamine synthetase serves as an entry point for reduced nitrogen, it is heavily regulated in virtually all organisms. The enzyme undergoes allosteric regulation by 6 end products of glutamine metabolism, including histidine, tryptophan, AMP, CTP, glutamine 6-phosphate, and carbamoyl phosphate, as well as glycine and alanine, which serves as indicator of the general status of amino acid metabolism in the cell. Each inhibitor alone produces only partial inhibition, but the effects of multiple inhibitors are cumulative, which provides constant adjustment of glutamine levels to match immediate metabolic requirements. Glutamine synthetase also undergoes covalent regulation. Tyrosine adenylation or the addition of AMP near the enzyme's active site increases its sensitivity to allosteric inhibitors. This process is regulated by an elaborate signal cascade. Tyrosine adenylation glutamine synthetase by adenylyl transferase is regulated by the P2 regulatory protein. The P2 regulatory protein is activated by tyrosine uridylation and inactivated by tyrosine deuridylation. The enzyme uridylyl transferase is allosterically activated by alpha-ketoglutarate, the precursor of glutamate and AMP, and inhibited by glutamine, which means that ultimately this cascade is activated by the reactants and inhibited by the product.